Plasma. This is the plasma. You see the universe? Now, this is, if you examine the plasma membrane, what do you first have to recognize? You have to recognize your bilayer leaflet, the two matrices that are stuck on top of each other. So you have, do you recognize the two leaflets, the upper leaflet and the lower leaflet? Do you see that? Number one. Number two, do you see your phospholipids? Yes, phospholipid, the head is the phosphate and the two tails are the lipid. The head is the phosphate and the two tails are the lipid. So they are inverted. Heads point to the outside with water, with the extracellular fluid. And the inside heads are pointing to the inner phospholipid. Heads are pointing towards the intracellular fluid, which is cytoplasm. Yes, water. The cytoplasm. The cytoplasm. So you see both heads are pointing to fluids that are polar. The extracellular fluid is polar. The intracellular fluid is polar. This is the two main things that you have to recognize. Then after that, when you look at this bilayer leaflet, what do you notice now? Do you see this huge protein that spans both transverses, both the outer and the inner leaflet? You see it, right? You see like a tunnel. So if things want to come from outside to the inside, they cannot cross this bilayer leaflet. They cannot cross this bilayer leaflet. They have to go through the channel. Who can cross this bilayer leaflet? Only lipids or cholesterol-based hormone like testosterone, cortisol, estrogen, progesterone, aldosterone. They can cross easily through the bilayer leaflet. Now, if we look, so these are one types of proteins. These are known as transmembrane because they cross both layers of the membrane. Are these the only types of proteins? No. There are proteins that are stuck to one side only, either the outer periphery or the inner periphery. Do you recognize them? These are known as, from their name, peripheral proteins. So you have to remember that transmembrane proteins cross both layers. Peripheral proteins are either on the outer periphery or the inner periphery. Another thing you must remember, these green structures, what are they? What are those green structures? Those green structures are sugars, glucoses. When you see green glucose with the lipid, what do we call that? Glycolipid. When you see the green glucose with a protein, what do we call that? Glycoprotein, because they're mixed together. So you see the sugars are always associated with proteins that are sometimes transmembrane proteins, sometimes peripheral proteins. But they're always where? Outside. Outside. Do you see any sugars down here? So glycoproteins and glycolipids are outside. On the bottom, do you see peripheral proteins on the bottom? Yes, right here. That's a peripheral protein. That's an example of a second messenger. Like those proteins that are polar, who cannot cross through, they bind to a receptor on the outside, then this second messenger peripheral protein is going to vibrate and do a intracellular signal transduction pathway, signal transduction cascade that is going to activate a second messenger, and the second messenger is going to activate some kinase, and the kinase is going to phosphorylate or target enzymes. Is that clear?